Yeah, thank you very much, Philippe. Thanks to the scientific committee for inviting me to this really great meeting. I enjoyed it a lot being here the last 2 days. So, as you can see, the institute I'm working at has a really long-standing tradition in working on colonization resistance. My group is interested in understanding microbiota-mediated pathogen resistance. And today I would like to tell you a story how we used a new approach to develop a defined microbiota that can confer colonization resistance against Salmonella. So I think it is really well appreciated that the microbiota has a very complex role in human disease and also human health. But I think it's clear that the functions of the microbiota are more beneficial rather than detrimental for us in general. So without a normal microbiota, we couldn't really survive. So this view hasn't always been like this. If you go back like 4,000 years to the ancient Egypt, they actually believed that the gut is home to many pathological processes, and they came to this idea because they observed that the food that they take in is actually rotting in the gut. It's not smelling very well, and for this reason, they actually very often used the technology of enema to treat many different kinds of diseases. And this has been really used until probably even now people use enema for treating many types of diseases in alternative medicine. So the idea has actually changed when we discovered that the gut is full of bacteria. And I would like to here mention the very famous physician Theodor Escherich, who also worked in Munich. He was a contemporary of Max von Pettenkofer. And he actually looked at the sequence of events that follow the birth of children. So he noticed already that the gut of babies is sterile, and then they become colonized by very complex bacterial communities. And he was also the first to describe the bacterium colon commune, which was later on named after him, Escherichia coli. And then he postulated already that there are good germs in the gut that can protect us against infection. So he was the first actually to promote the idea of colonization resistance. So what is colonization resistance? I think we have heard this very nicely already from Zhao. So the healthy gut is colonized by a very complex community, and this community mediates colonization resistance. When the community is compromised by antibiotic treatment, then the complexity usually is lower, and we refer to this state to dysbiosis. And a dysbiotic gut is very susceptible to various infections, for example, with very famous Clostridium difficile, but also Salmonella and various other opportunists. I would like to name here the antibiotic-resistant strains that cause severe problems in hospitals nowadays. So the dysbiotic gut is very susceptible to pathogen infection, and then pathogens induce antibiotic-associated diarrhea, which can also lead to chronic infections that are relapsing and that are the result of the microbiota that cannot recover from this infection or from this antibiotic treatment. And these infections can only be stopped when we transfer back a healthy microbiota by, for example, fecal microbiota transplantation. So what are the mechanisms underlying colonization resistance? So the mechanisms are as complex as the microbiota is probably. There are 3 different general classes of mechanisms. The first class, which I consider as a very important one, is interference, is exploitative competition. So bacteria actually compete with the pathogens for example, nutrients, for electron acceptors, for carbohydrates. The second mechanism is the interference competition, where the microbiota produces certain inhibitors, like bacteriocins, or certain metabolites, like short-chain fatty acids, or maybe even autoinducers. And the third mechanism is a rather indirect mechanism, where the microbiota or certain components of the microbiota can actually influence the immune system. So either they can inhibit or can stimulate certain immune responses, and those then are basically responsible or influence the susceptibility to the infection. So how can we actually, so far it's known that these mechanisms exist, but it's not really clear which bacteria are actually the main components that, or the main drivers of the colonization resistance. Actually, probably there are very many redundant processes going on at the same time. And one basically solution to this is to simplify the whole system. And we can do this, for example, by using gnotobiotic mice, which are germ-free mice that are colonized with single strains or a cocktail of gut bacteria. And these bacteria we can also grow very well in the lab, we can study in the lab, and then we can add or remove them selectively, and then we can actually address the relative contribution of these single bacteria to a phenomenon like colonization resistance. And the aim we always had in the lab was that we wanted to reconstruct a complex ecosystem with a simplified model that contains components of this complex ecosystem and that can recapitulate the colonization resistance. So we think about this as a model gut ecosystem where we have certain building blocks that we can remove and add, and we can build this entire ecosystem. And then we can actually define what of these components are really fundamental for the function, what components are essential only for part of these entire functions, and what components are just nice to have or even dispensable or redundant. So we were looking actually for candidates for our model system, and here I would like to mention the work by others that have actually shown that the microbiota is really host specific. So here's the work by Ruth Ley, and they have compared the microbiota of humans and mice for the first time in 2005. And here you can see that the composition of the microbiota is actually largely conserved at a high phylogenetic level, so at the phylum level, for example. You see that there in mice and in men, the most abundant phyla are the firmicutes and the bacteroidetes. But you can already see that some branches are only present in mice, whether some are maybe more enriched in humans. And there was also a recent study here by a consortium that actually looked at the mouse microbiome, and they compared the gene pool of the human and the mouse microbiome, and then it could show that only 4% of the genes are actually shared between human and mice. So we have to actually assume that the bacteria that colonize mice are really different from those that colonize humans. And for this reason, we really wanted to use mouse-derived bacteria for our mouse model system. So the current situation, when you go to strain collections like the ATCC or the German type culture collection, is like this. Basically, you find about a handful of bacteria that have been derived from the mouse gut. The only collection that is available is the Altic-Schädler flora. So this is a consortium of 8 strains that was isolated in the 1960s or 1970s by a researcher named Russell Schädler and colleagues. And they have been used ever since to colonize germ-free mice with a sort of healthy, defined microbiota that wouldn't actually harm the mice and that wouldn't contain any mouse pathogens, most importantly. The problem is that this consortium is protected by a patent, so it's not really freely available at the moment. Some labs have it, but it's not that you can just order it and work with it. So we decided we have to create our own collection of bacteria, and a PhD student in the lab, Sandrine Brugiroux, she started with this about 5 years ago and isolated bacteria from mouse feces, and the goal was actually to get the highest cultural diversity possible at this moment. So she isolated 60 different bacteria, and then she took out 12 that were derived from 5 different phyla. And now we analyzed them more thoroughly, and we found actually that among those 12 we already find 1 novel species and 2 representatives of a novel genus, so the red ones here, and also one representative of a novel family, which is we named this family Muribaculaceae, which is probably the family that is also now a candidate family as 24/7, which contains, for example, organisms like Barnesiella. So this actually reflects the situation in the current databases that there haven't been any strains derived from mice described so far. So if you go to mice, you can actually discover new families and new bacteria, and which is quite interesting, I find. So luckily, we were not the only ones who were interested in working with mouse-derived bacteria. And Thomas Clavel, who works also in Munich at the Technical University, he did a very brave study. He worked together with a German culture strain collection, the DSMZ, and they established a collection they termed the Mouse Intestinal Bacterial Collection, which at the moment contains 100 strains derived from mice, which are very well characterized. The genome sequence of most of these strains, and upon publication, they will be available to the public. So we also added our 12 strains, which you can see here. So they basically shows that they reflect a very broad taxonomic diversity when you look at the current culturable diversity of the mouse microbiota. Okay, so what we wanted to do first is to see how useful is actually this consortium that we selected. So the first experiment was conducted by Markus Beutler, also a PhD student in the lab, was to see what actually happens when you introduce this mixture of 12 strains into germ-free mice. And he followed the colonization kinetics of these 12 bacteria over time. And you can see that it changes during the first week, but then it adopts kind of stable composition, which is then maintained for at least 3 months. We also tested whether the consortium is actually transmitted from the mother to the offspring when you breed the mice for several generations in a germ-free isolator. And this is also the case. Here you see the parental generation, and then the mice were bred for consecutive generations, and the composition of this mixture actually stays rather constant over time. So there are no bacteria are actually lost. And the third experiment we did was actually we distributed this mixture also to collaborators, in this case here in Switzerland, in Bern and in Zurich. And so they also work with these communities. And we compared the composition of the microbiota in mice obtained from Zurich and from Bern with the Munich mice, and you can also see that the composition is the same. Of course, you have to use the same technology for analyzing microbiota composition, otherwise you get very different results. So what this tells us is actually that this community is very useful to generate also isobiotic mice, which are actually, for example, genetically different mouse strains that you want to colonize with the same consortium of strains. So now back to our initial question. So we wanted to have a model system which we could use to analyze colonization resistance in mice. And I would like to recapitulate on Andreas' introduction of Salmonella infection just very briefly. So Salmonella is usually taken up by contaminated food. And the first thing that Salmonella has to manage is actually to colonize the gut and to grow to high levels. And at this stage, it has to actually compete with the intrinsic microbiota. And in order to go to the second step, to invade the tissue, Salmonella has to reach a certain threshold levels in the gut lumen. So if it stays below 10 to the 5, it will never actually manage to go to the next steps. And the next steps are invasion of the tissue and replication in the underlying tissue, which leads to gut inflammation. And as we heard, this leads to Salmonella blooming, to disruption of the microbiota, and eventually the immune system, or this process is actually not really well understood, but inflammation resolves at a certain time and Salmonella levels go down. So we actually studied the first step of Salmonella infection and looked basically only at the colony-forming units in the gut or in the feces following oral infection. And here you see a summary of different types of mice, so colonized with different types of microbiota, when they have been orally infected with 10 to the 7 CFU Salmonella typhimurium. So here on the left side, you see normal conventional mice. These are actually 60 dots, and the majority of mice is not colonized, and some are colonized below 10 to the 5, so they will not develop symptoms of intestinal inflammation. By those 15% of the mice where Salmonella reaches sufficiently high levels, they will actually develop disease. When the mice are treated with antibiotics, for example, streptomycin or beta-lactams, then actually 100% of the mice get high levels of Salmonella, and they will also develop the disease. And the same is true for germ-free mice. Interestingly, we have shown previously that mice that are colonized with these with bacteria of the Altochedla flora also lack colonization resistance. So basically, from this, we concluded that actually ASF mice would be a good model system to test for any other bacteria that we could add on top of the ASF, to, whether they have the potential to mediate colonization resistance. So this was the first experiment we actually performed. We used mice colonized with the Altochedla flora, and then we performed fecal transplantation, either with a conventional microbiota or with our mixture of 12 on a control group that received no transplant. And then we waited for 40 days, and then took the mice and orally infected them with 10 to the 7 CFU of avirulent Salmonella that cannot trigger inflammation. And here we looked at the CFU per gram in the cecum 2 days after infection. And you can see that the mice that received the transplant, the fecal transplant from conventional mice, they are really protected. They have the transplant has restored full colonization resistance against Salmonella. While the Schedler flora mice, they are highly susceptible. And to our disappointment, actually, those 12 bacteria that we selected, they don't really, they induce some kind of colonization resistance, but it's not really impressive. So the next thing we did was we wanted to actually analyze what types of bacteria we have isolated, in order maybe to get a clue later on which bacteria from a normal gut microbiota might still be missing in order to make a complete and functional minimal gut microbiota. So we sequenced the genomes of the strains, and then we performed functional genomic analysis. So basically what you see here is a hierarchical cluster analysis of KEGG modules that are present or absent in the different strains. And here you see the strains that are color-coded. The green ones are the firmicutes. This one is the proteobacteria, and so on. And what you can actually see on the first side is that the functional classification of these genomes actually forms a very nice pattern that sort of reflects the phylogenetic membership of the strains, which is probably not that surprising. And we can see different clusters of genes or of functions. For example, here we see genes that are really conserved in the majority of strains. And here we see genes or pathways like the RNA polymerase, DNA replication, the sex secretion system, or ribosome. Then we have a cluster of pathways that is enriched in gram-negative bacteria, for example, biotin biosynthesis, LPS biosynthesis, and also outer membrane proteins. That also makes sense. And then we also see a cluster of functions that is enriched in gram-positive strains. And here we see lots of phosphotransferase systems, so sugar uptake systems, and other functions. So it's basically not really important what kind of function. I just want to say that this analysis really made sense to us. Okay, so the next thing we did is we wanted to know how many functions of a normal microbiota do we already capture with only these 12 strains. And for this, we generated an artificial metagenome from the 12 genomes of the oligo strains and compared it to shotgun metagenomes of conventional mice that we got from different sources. And you can see also we have a cluster of conserved genes, and we have some functions that are actually missing in our community. So we looked at these functions, and what you can see here that actually caught our attention first was there are lots of, I don't know if you can read it, but there are lots of cytochrome modules popping up, basically suggesting that so cytochromes, we heard just in the previous talk, are characteristic for bacteria that can respire, for example, facultative anaerobic bacteria or aerobic bacteria. So usually in the gut we don't have lots of aerobic bacteria, but we have a number of anaerobic bacteria, and those we have so far we haven't included in our strain collection. So we had the idea that maybe we have to add these facultative anaerobic bacteria in order to make the system more functional. So we had this nice resource in collaboration with Thomas Clavel, and we looked in the MyBC collection whether there would be some facultative anaerobes, and we found E. coli, we found a Staphylococcus xylosus strain and a Streptococcus strain. And we used those 3 strains, and we tested whether the supplementation of these 3 strains to mice colonized already with the oligomouse microbiota might restore colonization resistance. And surprise, actually really the 3 facultative anaerobic strains could restore colonization resistance to a normal level as mice colonized with a complex microbiota. And I have to say, if we supplement these 3 strains to mice with a Schädler flora, so this is data not shown here, but then it doesn't work. So they actually need the context of a complex anaerobic microbiota in order to mediate colonization resistance. So in conclusion, we have developed a defined mixture of 15 bacteria that can restore full colonization resistance against Salmonella. So currently we are trying to work out the mechanism, how these bacteria protect against Salmonella. So we speculate, of course, that they might compete for oxygen, which we just heard is very important for mediating colonization. So oxygen is very important for Salmonella to grow in the gut. Okay, so then I have also an outlook. I think the approach we used, we termed genome-informed design, and this might be also useful for actually looking at dysbiotic microbiota of patients and look actually which pathways are missing in this dysbiotic microbiota, and then go to a collection of bacteria and see which bacteria could be actually added in order to restore functionality and a complex gut ecosystem. And with this, I would like to end, and I would like to acknowledge all the people who were involved in the work. So there was mainly Sandrine Brugiroux and Markus Beutler and other people in the lab. And we had great collaborators, in particular David Barry and Alex Loy, Karina Pfann from the Department of Microbial Ecology in Vienna, Thomas Clavel from Munich, Cathy McCoy, Andrew McPherson in Bern, and also Sigi Hapfelmeier and Wolf-Dietrich Hart, who we shared the mice. Thank you very much for your attention.